right, so I'm Mrs. Murdock, and I'm going to explain something called gene mapping. And in order to understand gene mapping, let's first talk about the concepts here. Could you just close that door behind you? Okay, so let's just take a homologous pair here, okay? A homologous pair. And let's say that we're looking at two gene loci. Loci is plural for locus. All that means is the location on the chromosome of the gene, of the bit of DNA that codes for whatever protein it codes for. So here's one gene locus here and the other gene locus right there. So we're tracking how two genes are interacting specifically during prophase 1 of meiosis, which you may remember is when crossing over happens. So the thing about figuring out where these genes are on the chromosome, if you didn't know this, if you didn't have colored beads that popped up at you on the microscope, which they don't, how can you tell where these genes are in relation to each other? You already know they're linked, right? They're on the same chromosome. They don't follow independent assortment. How can you then map them? Well, it has to do with something called crossover frequency, or the frequency with which they switch places with each other. And that's going to start to make sense to you, right? So during crossing over, if they were nice and far apart like this, there'd be plenty of places for the chiasma to form. Remember the chiasmata forming? For those places to form randomly between those two genes, and for those either dominant or recessive alleles to switch places, right? So the further the distance between two genes, the higher the crossover frequency between them, okay? So there's a direct relationship. And the way you can tell whether crossover happens, remember that we usually expect a 9:3:3:1 ratio if independent assortment has happened. And if it hasn't, like let's say, here's the double dominant condition, you get a lot of those. Here's the double recessive condition, you get a lot of those, but you don't get so many of these, and you don't get so many of those. Let's say that instead of that expected ratio, you got like 200 of these, 10 of these, 10 of these, and 100 of those. I'll keep it round numbers, okay? This definitely doesn't fit 9:3:3:1, does it? So you immediately conclude, like with your fourth cross of flies, that, oh no, we've got linkage. They're on the same chromosome. Now the question is, how far apart are they from each other? What is this distance? The way you calculate that is actually pretty simple. These two types of offspring, here and here, are recombinant. They couldn't have happened unless the big A's and little b's crossed over and switched places. So the more recombinant babies there are, the more this happened between them, and therefore the greater the distance between the genes. So to calculate it, it's really simple. You add up the total recombinant babies, put it like this. So there's your recombinant babies, and you divide by all the total offspring, including the recombinants. Be careful of that. So 320. Okay, so 20 divided by 320, who's really good with their head? Oh, you guys are so good, thank you. Okay, so it's 16, or 0.16 would be what you'd get, right? And then you multiply that times 100, sorry, which, I'm sorry, did I not get that right?
You get 1/16.
So it's like 1 over 6.
Oh, 1/16. Do you know what that is in a decimal?
0.0625.
Thank you. So is it 0.06?
Yeah.
Can I do 0.07?
0.06 repeating, I think.
So round up to 0.07, right? Excellent. So then that would be 7 map units. I know, sorry if I'm doing bad with my arithmetic there. I'm great with concepts, terrible with arithmetic. All right, so there's my answer. That the distance between these two is, okay, we're just gonna say 7 map units. That's how you calculate it, okay? It only works with linked genes when you know they're on the same chromosome, when they're not fitting the expected Mendelian ratio that you calculated, okay? And then it's pretty simply, you just take the ones of which there are the fewest, because you figure those are the crossover events, add them up, put them over the total number of offspring, you get a decimal, you multiply by 100 to get a whole number of map units. And that's how you do mapping genes. But just to make the point, you see that if the yellow was much closer to the blue, it would cross over much less frequently. Can you see that? that if the yellow was up here instead of down here, right, then there's only one little place maybe, one little opportunity to separate them. Otherwise, if they separated down here, they would still be inherited together. And that is gene mapping, and I hope that was helpful.